Okay, so today we get to talk about genetic engineering. And so this will be a ton of fun because it's so cool and so many things are going on in the world with vegetables, good and bad, and a lot of fun things. So let's talk a little bit about it. So, with genetic engineering, the basic knowledge is used to derive applied science or useful products. So, like I said, it's kind of like a double-edged sword, and this might be a fun little discussion to have when we have class. There are direct, deliberate modifications of an organism's genome, and this is called bioengineering. Bio because it's dealing with the biology, engineering because we are changing it or designing it. And so combining this together with biotechnology, this is the use of an organism's biochemical and metabolic pathways for industrial production. So with tools and techniques of genetic engineering, we have the practical properties of DNA. And so these intrinsic properties of DNA hold true even in a test tube. So it's still always going to be double-stranded. RNA is always going to be single-stranded, etc. So there are some basic principles that will never ever change. Okay. When we do some of these techniques, the DNA is going to be heated from 90 degrees Celsius to 95 degrees Celsius. And basically what happens is this breaks some of those bonds, and they're like hydrogen bonds, right? And so then these two strands are separating because remember, DNA is a double helix. So when we do this heating, it will denature it and separate those two strands. And then we can replicate it, okay, and just target the piece that we're looking to make a copy of and transcribe it. And then when we slowly cool the DNA, this allows those complementary nucleotides, those letters A, T, C, G, to bond together with hydrogen bonds. And then the DNA again will form that double strand. Okay. Next slide. My computer's being slow. All right, so how do we do this? Well, we have enzymes for slicing and dicing and also reversing those nucleic acids. So we are going to use these restriction endonucleases, okay? And remember that proteins, specifically the enzymes that are specialized proteins, are going to end in the suffix of ase. And so they're also kind of named for what they do. So restriction endonucleases are enzymes that recognize specific sequences of DNA, and they break that phosphodiester bond that occurs between the adjacent or the nucleotides that are next to each other. Okay, and then the enzymes can be used to cleave or chop up the DNA at the desired sites. So it's not willy-nilly. We go in there with a targeted aim and then we chop where we want to. And then we're going to recognize and clip the DNA at the palindrome base sequences, which is the complementary base. And then we use this in the lab to cut DNA into smaller pieces. And this is called restriction fragments. Okay, so you can see that the cutting pattern is going to be very, very specific here. I'm cutting off some of your slides, guys. Sorry about that. All right, and so example here would be the examples of the endonucleases, the palindromes, and the cutting patterns. And the first two are staggered cuts, right? Where the third is going to be a blunt cut because this is staggered where it's cutting there and then cutting there, cutting there, and cutting there, where this is going to be a blunt cut and it goes straight through. And that is shown in figure 10.1. Okay, so basically the method in A can be used to separate the DNA strands lengthwise and make them single-stranded, where method B, which is here, is going to use the enzyme called those endonucleases to make the cuts of both across the DNA strands at specific sites. Okay. Right. Then we have restriction, fragment, length, polymorphisms. Say that 10 times fast, right? So basically DNA sequences vary even among members of the same species. That's what makes me and my sister, right? We're the same genetic base, but we have variation to make us different. And differences in the cutting patterns of these specific restrictions and endonucleases are going to give rise to fragments of different lengths. And those are called RFLPs, okay? So those enzymes that are used for dicing, splicing, and reversing the nucleotides, we also have one of those called ligase. And basically, this is a glue, right? So to ligate something means to stick it together or to glue it together. So this is where we're going to rejoin that phosphate sugar bond, which is that sticky end that was cut previously by those endonucleases. And then this is used for the final splicing of genes into plasmids and into chromosomes. So you can see here in figure 10.2 that these are examples of cutting patterns by restriction endonucleases and how they're useful in gene splicing. So in one up here, we have a restriction endonuclease that recognizes and cleaves the DNA at a site of the specific palindromic sequence. The cleavage usually produces staggered tails that are called sticky ends that accept complementary tails for gene splicing. This palindrome is cut by ACI1, okay, and that happens to be the enzyme that they're using. So down here in 2 then, the sticky ends can be used to join the DNA from different organisms by cutting it with the same restriction enzyme, ensuring that both fragments then are going to be 2 complementary ends. And this, for instance, this palindrome, right, this complement is going to be cut by Taq polymerase or Taq1, okay? So that is that picture, and again, that is figure 10.2. So then we have reverse transcriptase, and this makes a copy of it makes a DNA copy of RNA, which is called cDNA. And cDNA can be made from either the mRNA, the tRNA, or the rRNA, which are types of RNA that we talked about last week. And these provides a means of synthesizing or making eukaryotic genes from mRNA transcripts. And these are synthesized genes that are free of introns. And remember from last week, those introns are like the junk DNA that we're actually finding have some information in it. Okay, so this here is figure 10.3. And this is what we do when we synthesize cDNA from eukaryotic messenger RNA. So starting here up at the top, the bacterial cloning host cannot process intact eukaryotic genomes. They're too big. So to provide a gene that will transcribe and be translatable by a bacterium, first, up here in A, we have to modify that messenger RNA, which again lacks introns, and it's isolated from the source in the eukaryotic cell. Okay, so we're removing those introns. We have it located here, okay? So then in B, this strand is going to be converted in a test tube, because remember, this is happening outside of the body, to a single-stranded DNA by reverse transcriptase and a double-stranded DNA by a polymerase. And then this copy of DNA that is named cDNA can then be transcribed and translated by the bacteria. Okay, so that's what's going on in that figure. And again, that's figure 10.3 in your book. Then we get to analyze it. So how do we do this? Well, one of the most popular ways to look at this would be through gel electrophoresis. And so what this does is it runs a current down through your samples, and it separates the DNA fragments based on their size. And the DNA samples are placed on a soft agar gel, and then they're subjected to an electrical current to help separate those guys out. Okay, so this is figure 10.5, and up in A, which you can see right there, this is where a restriction endonuclease is applied to the sample using, sample 3 as an example. And then the sample of the DNA fragment is loaded into the well at the top of the gel and undergoes electrophoresis. Then the final position of the fragments will be visualized by staining or the method shown in B, which is going to be coming up in a few pictures. Okay, so basically what we're going to be doing is this restriction endonucleases selectively cut the sites of the DNA, and then we have the different fragments. We're going to put them in the DNA, in the agarose gel. We're going to shoot electricity through it, and you can see it's going to go from the negative down to the positive, and we're going to separate those by size. Okay, and then the negative charge of the molecule causes the DNA to move towards the positive pole, and then this rate of movement is dependent on the size of the fragment. Think about this. If you're trying to push a boulder through like Jello, because like agarose gels kind of have the consistency of Jello shots, right? They're thicker than Jello. They're kind of thick, but it's gelatinous, right? And so if we try to push a boulder through it, smaller ones are going to go faster. Larger ones are going to take longer times. Okay, so the rate of movement is very much dependent on the size of the fragment. And then this would be part B of figure 10.5. Okay, and so with this, a developed gel reveals the separated bands of DNA fragments. The size of a given DNA band can be determined by comparing the distance traveled to the distance traveled by a set of DNA fragments known as a size ladder. Okay, and that's in this one, lane 5. So that's the molecular marker. Okay, And then this is measured in the number of base pairs. And then the gel contained DNA with radioactive bases that can expose a photographic negative. So basically, you can put a tag on this. Okay, usually we use ethidium bromide. Okay, cancer causing, but it's nice and purple and it's pretty. And you put it under UV lights and it fluoresces. Okay, so you can tell a lot of things from an agarose gel. Okay, you can tell the size as well as amount that's there. So looking at more of these methods for analysis, we have nucleic acid hybridization and probes. So this is when single-stranded DNA can unite with other single-stranded DNA or RNA, and RNA can unite with other RNA, and this is called hybridization. And so this is what we use for foundation of gene probes. And these are short DNA fragments of a known sequence that will be base paired with a stretch of DNA from a complementary sequence, if one exists in the sample. This is useful in detecting specific nucleotide sequences in unknown samples. So another way that we can look at these is using a southern blot. Personally, I've never done a southern blot, but they're very cool. And so southern blots, I've done western blots, okay, and there is no eastern or northern. So I don't know why. But anyway, it is what it is. So DNA fragments are separated by electrophoresis. They're going to be heated to be denatured, and then they're going to be incubated with specific DNA probes. Then these probes are going to attach to a complementary segment if that complementary segment is present. And then we can use this to isolate fragments within a mix of fragments and find that specific gene sequence. Okay. So looking at your southern blot, this picture is going to be in your book as well. Let me see. I'm going to pause you so I can find it. Hold on. Okay, so I couldn't find the picture in your book, but basically with southern blots, it's going to be kind of the similar thing where the samples are cut with the restriction enzymes. It's going to be denatured and it's put in a gel. You have a specialized media with what's called buffers so that can help with the electrical charges. Then we're going to separate them and expose them to the X-ray film so that we can then look at the fragment sizes and locations. Okay, so it's actually pretty cool. Alrighty, so next slide. So looking at other methods, we can also use the FISH method. And this is fluorescent in situ hybridization. And this is when probes are applied to intact cells, and then they're observed for the presence and location of specific sequences. And this is very useful in genetic diagnostics, okay? And so this is useful for locating genes or a location on a chromosome, okay? So it's very cool. So then we have DNA sequencing, and fun fact, after my postdoc, my first job was running a DNA sequencing facility, so I happen to know quite a lot about this, okay? And so basically with DNA sequencing, we can determine the actual order of the types of base pairs for all types of DNA, and this means ATCG. So I can run a gene through my sequencer and I can figure out its genetic code, okay? So when the Human Genome Project was huge back in the, was that early 2000s, right? I almost just saw. It was a really big deal and it took a lot of time. And so the most common sequencing technique is called Sanger sequencing, okay, or the Sanger techniques. And this is when the test strands are denatured. They're heated enough that they unravel that double strand to serve as a template to synthesize or make complementary strands. Then the fragments are going to be divided into tubes that contain primers, DNA polymerase, all 4 nucleotides, which would be your ATCG, and then fluorescently labeled DNTPs, so ATs and Cs and Gs. Okay, and so this is pretty cool. And so with Sanger sequencing, we don't have this in your book because it's actually very cool. So the DNA is going to be sequenced, you denature it so that it is going to be separated. Oh, here we are. This is in figure 10.7 of your book. So some basic techniques in DNA sequencing. So the sequencing of 9-based strand using the Sanger sequencing. The sequence in number 6 is read from the bottom to the top, and it must be converted into its complement to get the true nucleotide order of TCG, GCT, AGG. And then, so that was steps 1 through 6. And then in step 7, So you can read through these. Okay, so basically the DNA sequence, it's denatured, so we can expose a single strand. We put a primer that's specific for the gene that we want to amplify it. We put it all in a test tube with fluorescently labeled nucleotides. Okay, and then during the replication, each new strand is going to be terminated when they get to a tagged letter. Okay, so they'll have regular A, T, C's, and G's in there, and then they'll have tagged A, T, C, G. And basically, it's kind of like a big balloon on this tag, and so it gets too big. And so once it hits one of those balloons, it stops replicating it. Okay, and then basically at the end, you run it through a very, very thin copper tube, right? And it separates it by size. And so the smaller ones are going to get there first, and then a laser actually looks and it measures the color. So it knows that when it's green fluorescent, that would be an A. It knows that when it's yellow, that that's a G. When it gets to blue, they know that it's a C. And so based on that, you can look at this genetic sequence and you can figure out the ATCG order. So using methods to size, synthesize, and sequence DNA, studying the genome of an organism has created many, many, many new fields in biology. We have genomics, proteomics, metagenomics, and metabolomics. Okay, so genomics is the study of an organism's genes and their functions, where the proteomics is taking it a step further and looking at the proteins and then the functions that are created by those proteins. So when I was in grad school and when I was running my own labs, I was a proteomics girl because I studied protein expression, right? Metagenomics, this is the study of all the genomes in a particular ecological niche, as opposed to individual genomes for single species. So we're looking at the broader picture. And then metabolomics, this is the study of complete complements of small chemicals that are present in a cell at any given time. So then we move to another very important test, okay, and actually hospitals use this for genetic screening, to confirmatory tests for diseases, things like that. And this is called PCR, and that stands for polymerase chain reaction. And this is a method that we use to amplify DNA. And this rapidly increases the amount of DNA in a sample, which again is in a test tube, okay, so it's under a very controlled environment. So primers of a known sequence are added. So say you are testing a patient for the presence of BRCA, okay? So you would put a primer in that test tube that's specific for BRCA1, okay? And this would indicate where amplification would begin, along with special heat-tolerant DNA polymerase, which is called Taq polymerase. Fun fact, they found it actually in a hot spring, as well as those nucleotides, which are your A's and T's and C's and G's. And then this is repeatedly cycled through denaturing, which is separating that double strand, the priming, which is attaching that primer to the part that it complements, and then extension. So that would be making the new DNA strand, is specifically what you're looking for. And then each subsequent cycle is going to double the number of copies for analysis. So if we start cycle 1 with 1 copy of DNA, it will double. So at the end of cycle 1, we'll have 2 copies. Well, then that 2 will double for the second cycle, and you'll end up getting 4. Then you double it again for the third cycle and you get 8. And then you double it again for the fourth cycle, you get 16, etc. And this is essentially very important in gene mapping, the study of genetic defects in cancer, as well as forensics, taxonomy, and evolutionary studies. Okay, so this is figure 10.8 in your book, and this is the diagram of what happens with the PCR reaction. And so this is just showing you the details of what's happening over 2 cycles, because we are doubling at each time. And so this is going through those the cycles. Cycle this one is the DNA sample, then this is the denaturation where the strands are going to separate, and then you have the priming. And this is where the oligonucleotide primers attach to the ends of the strands to promote replication and amplification. And then here you have extension. And that's where that Taq polymerase, which is that heat-stable DNA polymerase, is going to synthesize or make a complementary strand. Okay, and then after that, we cycle it through again, and we have denaturing, priming, and then extending. Okay, so you can see all of that, and then this is going to exponentially grow, right? It is literally a logarithmic exponential graph. It doubles each time. Okay. So moving on to recombination of DNA. This is the intentional removal of genetic material from one organism and combining it with that of another organism. And the objective of this recombinant technology is cloning, and this requires that the desired donor gene be selected, excised or cut out by restriction endonucleases, so a specialized enzyme, and then isolated. And then that gene is going to be inserted into a vector, and this vector is usually going to be either a plasmid or a virus, and then that will insert the DNA into the cloning host. Okay, then the cloning host is usually going to be a bacterium or a yeast that replicates quickly and is very controllable, and the gene then can translate it into a protein product. So this would be an example of your recombinant DNA techniques. Right, so let me pause and find this. This is figure 10.9, which is the overview of the methods and application of genetic engineering. Okay, so the practical applications of genetic engineering include the development of pharmaceuticals, genetically modified organisms, and then identification techniques. Okay, so hold on a second. All right, sorry, I have a reminder on my computer and personal information was going to pop up, so I needed to pause for a second. Okay, so this is again figure 10.9. So basically what's happening here is that human or other mammal, microorganism, or plant cell, it's going to be here, and then we're going to take the DNA of interest that's isolated and produced from mRNA, and then the DNA is going to be inserted into the vector, right, which is just like an endless circle here. And then the microbial cloning host is going to be transformed when it picks up the vector with foreign DNA. And then the multiplication of cloning is used to, of the cloning host is used to amplify the protein, and then it's transformed into the process of foreign DNA into the protein. And so an example of what this is used for is in pharmaceuticals. Okay, we can produce hormones this way, and one of them would be insulin and human growth factor, as well as vaccinations like hepatitis B, and also for industrial chemicals. Where on the other side here, we can branch off, and then the growth is going to be transformed to cell supplies DNA to study and engineer other organisms. And so we can use these altered organisms here for, you know, economically useful traits, so pest-resistant plants and herbicide-resistant plants, improved nutritional values, etc. Or we can also have additional multiple applications in genomic research, testing and therapy, such as gene function, nucleotide sequencing, identification, gene libraries, and gene therapies. So again, this is figure 10.9 in your book. All right, so what are the characteristics of these cloning vectors? Well, they must be capable of carrying a significant piece of donor DNA, and they must be readily accepted by the cloning host. And so these plasmids, which is that little circle guy, these are small, well-characterized, I said that was vectors. Vectors and plasmids are very similar, but they're well-characterized, easy to manipulate, and they can be transferred into an appropriate host cell through the process of transformation. Where bacteriophages, these have the natural ability to inject their DNA into the bacterial hosts through transduction. So when we are looking at vectors, there are a couple things that we want to consider, and this is figure 10.10. And this is a partial map of the PBR322 plasmid of E. coli. And so the arrows that you can see on this figure are going to delineate sites, point out sites. They like to use really fancy words. Okay, delineate is just another fancy word for like point out sites that are cleaved or cut by various restriction endonucleases. And the numbers indicate the size regions in kilobases, which is abbreviated KB. And then other important components are genes for ampicillin and tetracycline resistance and the origin of replication. Okay, so the origin of replication is needed so that it can be replicated. And then the vector must accept the DNA of the desired size. So we have to make sure that it fits. We can't put like an elephant in a room where goldfish is supposed to be, right? It has to be the right size. And then the size, and then the gene, which confers to the drug resistance to their cloning host, is going to be the one that is going to be amplified and inserted in here and be able to grow. Okay. So when we're looking at what is desirable features, this is table 10.1, and this would be that they can grow really fast. They can be grown in large quantities. They are not disease causing, right? Because that's kind of important. The genome is well mapped, okay, or delineated. It's capable of acquiring plasmids or bacteriophage vectors, and it also maintains foreign genes through multiple generations. And we also want it to secrete a high yield of protein from the expressed foreign genes. So looking at this is figure 10.1, the part of it, the first part of it. And so how do we do this? Well, we prepare the isolated genes for splicing into a vector by digesting the gene and then the plasmid with the same restriction endonuclease enzymes. And this creates a complementary sticky end. Remember the areas that we can glue together on both the vector and the DNA that we want to insert. Okay. Then this gene plasmid pair are placed together, and their free ends are going to base pair, and then that glue, that ligase, remember to ligate is a fancy word to bind, glue together. The ligase is going to join those two ends together, and then the gene and the plasmid combination is called recombination because it's not the original combination, it's recombinated, right? And so that is what is happening. And again, this is explained in figure 10.1. Well, then the recombination or the recombinant DNA is then going to be introduced into a cloning host, and then the cloning host is going to transcribe that gene and translate the protein. Okay, and so this is where you can see the complementary strands here in 4. I'm reading from here and starting here. The complementary ends are going to be bound by that ligase into a single recombinant plasmid, and then we undergo transformation. I didn't realize that was going to move. And then this recombinant plasmid is going to be introduced into the competent cloning host bacteria, and the cells cultures are going to be grown on media that select only the clones that are antibiotic resistant, therefore carrying that donor gene. And then down here in 6, you can see that the recombinant isolates are grown in culture to increase the numbers, and then every cell will now contain a replicated plasmid that is going to be carrying that gene. Then down in 7, this is where the recombinant cells are induced to transcribe and translate the donor gene, and then the final step down here in 8 is going to be that this is going to yield a protein product which can then be recovered from the culture and purified, giving you what you were looking to isolate. Okay. So we can use this to screen for recombinant microbes, okay? And this is figure 10.12 in your book. And this is how we can use selective media that we talked about in one of the very first lectures to quickly identify those recombinations, okay, the recombinants. And so up here in A, which is over, it's labeled 1 here, the plating of the culture on a non-selective medium will not separate the transformed cells from normal cells because these, which left the plasmid. So that one's not going to be a very good isolating media because everything's going to grow. Or over here in B, or 2, plating on the selective medium containing, for instance, ampicillin will permit cells that are, only the cells that are containing the plasmid, ampicillin, like antibiotic, right, to multiply. And so the colonies growing on this medium that carried the clone gene can then be used to make a culture for those gene libraries, also for industrial production and other processes. Okay, so you can see that by selectively growing only those that we want, then we can isolate what we want. So biochemical products of recombinant DNA technology, this whole skill is going to enable large-scale manufacturing of life-saving hormones, enzymes, and vaccines. One very big example is going to be insulin, okay? So we can use this to grow insulin to treat diabetes, okay? Also, human growth hormone for dwarfism, erythropoietin for anemia, factor 8 for hemophilia, and the HBV vaccine. So there's a lot of really good things that can come out of this technology. This is a summary of table 10.2, and these are examples of protein products that are that have come from the recombination of DNA. So you can see that there are a lot of really awesome things that keep our society functioning as a whole. So moving on to genetically modified organisms then, these are recombinant microbes so that we use. Okay, so for instance, Pseudomonas syringae, these prevent ice crystals. Bacillus thuringiensis, this can encode for an insecticide. So many enzymes, hormones, and antibiotics that are used in drug therapy, that are used in drug therapy are manufactured using mammalian cell culture. and cell cultures can be used to modify those proteins. And so from microbes to the bioremediate disturbed environments, we can do this to improve them. And then you can also have oncogenic adenoviruses. And these host range, the host ring consists of cells that produce cancer-specific proteins. So we can use this for a lot of very important things. So looking at transgenic plants, this is figure 10.13. So we can use Agrobacterium, which is a natural tumor-producing bacterium, and then the Thai plasmids insert into the genomes of the infected cells. So you can see starting up here at A and going down, the large plasmid TI of this bacterium can be used as a cloning vector for foreign genes that code for the herbicide or the disease resistance. And then down here in B, the recombinant plasmids are taken up by the agrobacterium cells, which multiply and therefore copy that foreign gene. And then down here in C, the genetically engineered agrobacterium is going to be inoculated or injected into a culture of target plant cells, and this would then infect the cell. Down here in D, then the bacterium is going to fuse with the plant cell walls and the TI plasmid enters. And then the tDNA is going to carry that herbicide gene into the plant nucleus. And mature plants can be grown from single cells, and these transgenic plants will express the new gene. And then finally, down here, because the gene will be part of the plant's genome, it can be transmitted to the offsprings by the use of seeds. Okay, so we can use this a lot, and it can be very, very helpful. So here are some examples of transgenic plants, and this is in figure 10.3. So we have cherry tomatoes, wheat, rice, soybeans, watermelons. You can engineer for just about anything that you want to these days. Okay. So looking at this with transgenic animals then, this is figure 10.14. Okay, and this is when we use a virus to transfect a fertilized egg or an early embryo. And then these transgenic animals will transcribe and translate the eukaryotic genes, and then the transgenic animals will transcribe and translate those. Oh, it says the same thing. Okay, so basically several embryos are going to be recovered from the female. Okay, then the embryos are going to be transferred to a depression slide, and so that's a slide that has like a little divot in it, and then that divot contains culture media. Okay, and then the embryo is going to be held in place, and then the DNA is injected into one of the pronuclei. Okay, and then this is just showing you the DNA injection, and then several injected embryos are going to be placed into the oviduct of the receptive female, and that way we can kind of take over the body and grow what we want to. Okay. All right, so animal models have been designed to study human genetic diseases. You can have mouse models for cystic fibrosis, Alzheimer's, sickle cell anemia. I used to study hydrocephalus on a rodent colony, and so these ones spontaneously developed hydrocephalus, and so I could use them to study their brain and how I could fix their brain, right? We could also use sheep and goats to manufacture proteins and excrete them so that we can do high scale. So some examples of transgenic animals are here, and so this is figure 10.4. You can see that we have mosquitoes, we can have goat and silk fibers, you can have pork for meat processing, mice, cats, pigs, salmon, cows, right? There's a lot of different things that we can use for the greater good, okay? So moving on to genetic treatments. So we can take this and introduce DNA into the body, and this is where it can kind of get gray. When you have a good heart and you're doing it for good reasons, this is usually pretty good. But unfortunately, there are always people that are not for the good, right? And so, we just kind of have to keep our morals in check here. So for gene therapy, which is good, we can correct and repair a faulty gene in the humans. And so one example of this would be ex vivo therapy, and this is where the normal gene is cloned in vectors, then the tissue is removed from the patient. Where in vivo therapy is where the naked DNA or vector is directly induced into the patient's tissue. Okay, so again, you can see over here in this figure, the normal gene's isolated, gene's cloned, it's inserted into the vector, bone marrow sample is taken. All right, then the stem cells are going to be transfected, and then you're going to put it into the patient and you're going to observe it for expression of the normal gene. Okay. So using DNA technology as genetic medicine, well, there are gene silencing techniques. Okay, so there's an antisense RNA, and this is where it has the bases that are complementary to the sense strand of messenger RNA. Okay, and this results in a loss of translation to the messenger RNA. We also have antisense messenger RNA, and this is where DNA, and this is where it is delivered to the nucleus. Then it's going to be bound to the specific messenger RNA, and this is going to be blocking the reading of messenger RNA in the transcripts on the ribosomes. Okay, so again, we can do good things and we can do bad things. Hopefully we will all stay in the good stuff, right? So wrapping up this chapter, looking at genome analysis, we have a thing called DNA fingerprinting. And this is where every individual has a unique sequence of DNA. And methods used to do this include restriction, endonucleases, electrophoresis, hybridization, and the southern blot that we talked about earlier. So looking at this lovely picture, we can see what they are talking about. So let me try to find this in your book. Let me pause. Hold on a second. Okay, couldn't find this figure, but basically, this is an example of how we can use it for good, right? So we have cells from different samples that are processed to isolate their DNA. Then the DNA samples are exposed to the endonucleases, which are going to cut them into specific sites on a series of different fragments. And then the electrophoresis or separation of the DNA fragments is going to help separate them by size, with larger fragments near the wells. Although they're invisible to the naked eye, when we use the ethidium bromide or other techniques to visualize them, then we can fluorescently tag them and look at them under UV lights. And so this would be an example of an actual DNA fingerprint that was used in a rape trial. So the control lanes with known markers are in lanes 1, 5, 8, and 9, where the second lane is going to contain a sample of DNA from the victim's blood. The evidence samples 1 and 2, which are in lanes 3 and 4, right? They contain semen samples that were taken from the victim, and then suspects 1 and 2, which are in lanes 6 and 7, were tested. And so by comparing these, we can tell which suspect, which would be suspect 1, would be matching with the evidence, because you can see the markers are the same. Okay. Alrighty, so then again, looking at this genome analysis, there are different types of analysis. One of them would be SNP, which is a single nucleotide polymorphism, as well as looking at your other markers such as VNTRs and the microsatellite polymorphisms. So basically fancy words to say we can catch bad guys when we use this for good things. So don't do bad things. All right, DNA fingerprinting. This is used to identify hereditary relationships, and we can use this to study inheritance of patterns of diseases, also human evolution, and also to identify criminals or victims of disasters when we can't use fingerprints, right? And then the analysis of the mitochondrial DNA is used to trace evolutionary origins, because mitochondrial DNA comes from the mother. Then final slide, we have the microarray analysis, and this is taken similarly off of figure 10.17. Did this in grad school for my PhD. It was when it was brand new, and it was actually kind of fun to do. So the microarray analysis is going to track the expression of thousands of genes, and this can be used to identify and devise treatments for the diseases based on the genetic profile of that disease. And basically, this screening analysis helps scientists to identify which genes we want to study more closely to figure out which ones are actively being expressed in the cell. All right. We'll see you guys in class. Thanks. Bye-bye.